So welcome everyone to the Build-a-cell chemical reaction network modeling series. This is the first lecture we have out of 6. I am William Poole. I'm a graduate student at Caltech working with Richard Murray and Eric Winfrey, and I'll be talking about and giving you guys an introduction to what chemical reaction networks are and how to build them using the biocompiler software. So first, to give you a little overview of this series, this is the first lecture where we're going to really introduce what CRNs are at a high level. The next lecture will talk about how you can compile chemical reaction networks automatically so you don't have to build them by hand. In the third lecture, we'll talk about combining different chemical reaction network models together with the sub-SBML software, and also transports between compartments. In the fourth lecture, we'll talk about stochastic chemical reaction network models and simulation. In the fifth lecture, we'll talk about how to connect chemical reaction network models to your data using Bayesian parameter inference with the bioscrape package. And in the sixth lecture, we'll talk about how you can scale up chemical reaction network simulation and interface chemical reaction networks with other simulation modalities using the Vivarium software package. And as you'll notice, there's a number of co-instructors. Ayush Pandey and Iran Agman are both teaching some of these lectures as well as me, and we have a very amazing team of TAs who will be helping you throughout all of this, who I will introduce at the end. So I want to start by asking, why are you here? Why do you want to learn how to do CRN modeling? And this will also give me a moment to talk a little bit more about the particular software we use. So as a synthetic biologist and an engineer, I often talk about the design, build, test cycle. And what does this mean? Well, designing is, maybe using a computer, ideally, to help you design how a system should work to achieve the results you want. And we're not really going to be talking about design specification tools, insofar as to say that this is an active area of research that many of us are very interested in. What we will talk about is how to design things by hand. And this is where model creation comes in. And biocompiler and sub-SBML, which will be mentioned in the first 3 lectures, are 2 tools to make it very easy for you to build complex and modular chemical reaction network models that you can scale very easily. Once you have a model, you have to test that model. And part of sort of the model building process also requires you to simulate the model. And so we're going to be using the bioscrape simulator, which comes out of the Murray lab as our default simulator. And in week 4, we'll talk about stochastic simulation using bioscrape lineages. There are many other simulators out there. I've given you a list here. And if you follow the link on the bottom, there are many more simulators you can use. And what's cool about our software is that biocompiler and sub-SBML both use this file format called SBML, which allows them to interface with many different simulators. You don't have to use our simulator, although we'll be using it because we know it best. Once you simulate a model, you might decide to do some sort of mathematical analysis of the model. And that's something we're not really going to be talking about, but there's many analysis tools. And this is a way to test your model. Does it behave the way you expect? Is it stable? Is it robust, etc.? Instead, we'll talk about a different way of testing models, which is how you test them with data. And one way to do this is by using bioscrape inference to do Bayesian parameter inference to parameterize your models, in other words, find parameter values, given your experimental data. This is a really important process because once you can connect your model to data, you can go ask, is my model behaving the way I expect it to based upon my data? And that will inform your next round of design, building, and testing. There's also sort of a biology pipeline here that's less about building things and more about understanding hypotheses. So you might start with some data that you've collected over the years, and you can use model building with biocompiler and sub-SBML as a way of mathematically codifying your hypotheses into something very concrete. A model is a collection of hypotheses that you've written down. When you do parameter inference, you're connecting your models rigorously to your data. You're asking, can my model explain my data? And can my data parameterize my model? In other words, is my data even informative enough to explain or to parameterize the complexity of my model, or is my model complex enough to explain the complexity of my data? And based upon that analysis, you can do this iteratively, go back and produce more models until you get a model that really seems to match your data very well. And that model might represent your best current hypothesis. And after going back through this hypothesis testing loop many times, which is cheap and fast because it's all via computers, you then go say, okay, I have this best hypothesis I've come up with using my data and representing my hypothesis as a model. I can now go do more experiments, which is going to be much slower and more expensive, to get even more data to then test that model and refine it more. So the idea here is to use models to help you design experiments so you can use your lab time and resources more wisely. So in all of this, I've sort of thrown around the word chemical reaction network a bunch of times, and I've talked about models. Chemical reaction networks are the models we're going to be using in this course. And they're a very widely used mathematical model in synthetic and systems biology. I was just Googling chemical reaction network models in Google Scholar. If you type in chemical reaction network, there's over 5 million papers that come up in patents. They're very widely used across many disciplines. So let's define them real quick. So we're going to see a little bit of math here. We have a set of species, which I'm calling x1 through xn, but these can be anything you want. They could be genes, they could be molecules, they can be atoms, they can be abstract symbols. I'll give you some examples in a second. You then have a set of reactions where your input species become output species at some rate or propensity for each reaction, which is a function, typically a positive function. Chemical reaction networks are expressive, so they take many forms. For example, I might have sort of a more traditional chemical reaction network from chemistry where oxygen and hydrogen form together to produce water. This is when hydrogen burns. And here my species are really atoms or gas molecules that are changing bonds in the reaction. A more abstract example from biology might be that I have a gene which produces a transcript. And maybe here my rate function is a function of the amount of polymerase or some transcriptional factors that matter. This is much more abstract. Here I'm actually talking about a whole process where there's polymerization of this transcript as it's copied into RNA. I'm taking that very complex process and saying, okay, well, at a high level, this is just a gene producing a transcript. And I can make this even more abstract. And I could imagine, that I'm not even talking about molecular species anymore, but I'm talking about real species like rabbits. And maybe I have a white rabbit and a black rabbit who come together to produce 5 new gray rabbits. And this can also be represented as a chemical reaction network. And here I'm not talking at all about how this happens. I'm just saying, when rabbits come together, more rabbits happen to appear, I guess, in increments of 5. When you write out these reactions, they end up becoming ordinary differential equations, at least in the deterministic semantics. So what does that mean? Well, I will introduce a little bit of notation here to show you how you actually get differential equations from a CRN. So this is the same inputs to outputs I showed you earlier. Maybe this, but here I'm using a different notation. So I sub I superscript R is saying how many of input I are there in reaction R. And O sub I superscript R is how many outputs of species I are there in reaction R. So in the rabbit example, I would have 3 different types of rabbits, black, white, and gray, and my inputs are 1 for black and white rabbits and 0 for gray rabbits. And my outputs are 1 for black and white rabbits and 5 for gray rabbits. And then you have your rate function at which this happens. So this is just a mathematical notation. You can think of each of these as a vector over our species, where I'm having a species input vector go to a species output vector. And this lets me write down a dynamic equation as the form of a differential equation. So the change in xi with respect to time is equal to the sum over all my reactions of the outputs minus the inputs, the change of species I due to that reaction times the rate that reaction occurs at. So this is a pretty simple differential equation formula. These can get very complicated when you actually write them out. And there's also stochastic semantics, which I'll talk about in a later lecture. So to give you an example, Here is a chemical reaction network with 3 reactions. The first reaction is nothing goes to A at rate 10. Then there's a reverse reaction, A goes to nothing at rate 2A. So I'm writing out the exact rates here. And then I have C plus A goes to C plus 2B. So C is catalyzing A to become 2B, and the rate that that's happening is C times A divided by 3. So if we write out this differential equation, well, we first see that the change in the catalyst C is 0 because C is not involved in these reactions, so that doesn't do anything to it. And then in this case, C is both consumed and produced, so its net change is 0. So when you write out this OI minus II, you get 0 regardless of the rate. Well, if I look at B is only produced from one reaction, it's never consumed, and 2B are produced from this reaction, and this reaction occurs at rate Ca over 3. So I end up getting 2 times the rate of the reaction Ca over 3. A, on the other hand, is produced and consumed in 3 different places. It can be produced from nothing at rate 10, so I have a positive 10 here. It can also be degraded to nothing at rate 2A, so I have a 2A here. And it can also interact with C to produce B at a rate Ca over 3, so I have Ca over 3 here. Note that A is always produced and consumed in units of 1, so I have sort of hidden little ones out here for the change in A from each of these reactions. And if you simulate this, it ends up looking like this. So A goes down, C remains constant, and B goes up, which is what you might expect. Now, when you're thinking about chemical reaction network modeling, understanding the differential equations is not super important because we have simulators like Bioscript that will produce those differential equations for you automatically. What's really important is thinking about chemical reaction networks as a language to represent biological concepts that you think about all the time. And so I'm calling these motifs. These are common little chemical reaction networks that you use over and over again and get combined together to produce larger, more sophisticated models. So the first motif is binding. And binding is when you have 2 molecules, A and B, which are maybe floating around and bind together to form a complex, A:B, which is a complex of the things bound together. The second motif is catalysis. This is where some catalyst C changes A into B. And exactly how this happens can be very abstract, but the key thing about catalysis is the catalyst isn't used up. So enzymes are catalysts. You can also imagine compartments. So here I have A in 2 different compartments, internal and external. I can diffuse between them. And this can be represented as a very simple reversible chemical reaction network. Finally, you might have some set of discrete states. Maybe A has 3 states. It's a protein. It can be open, it can be in a transition state, or it can be closed. And I can represent the movement between these discrete states as a chemical reaction network as well. Another example of sort of states might be phosphorylated versus not phosphorylated for a protein. Using these motifs, we can build very large networks, as I'll show you in a few slides. The one thing I haven't shown you are the rates. So that's what we're going to talk about next. How do you choose the rates that these reactions occur at? I guess before we get into rates, I have to tell you what CRNs cannot model. This is very important. You want to make sure that you're using them for what they're good for, but you're not trying to use the CRN framework to model something that's really probably not going to work. And the key thing CRNs can't model is the location of molecules in space. What do I mean by that? In the mathematical world, we say CRNs are well mixed. Everything is sort of in the same location. So that means if you have complex spatial structure, we can't really use CRNs to model that. So for example, continuous motion. So here I have, I'm showing you diffusion in 2D, where every single particle has an X and a Y position as well as a velocity. CRNs would not be good at modeling this at the level of each particle because we are not keeping track of where particles are located. Everything is assumed to be well mixed. Another example are detailed chemical structures. For example, this alpha helix I'm showing you here is a protein secondary structure where maybe you care about the location of all of these atoms and molecules and how they are aligned relative to each other. For complex 3D structures like this, chemical reaction networks are not the right model, again, because they don't really have the locations of molecules in space. Everything is well mixed. I could talk about this alpha helix as a whole unit and maybe that it bumps into something else, but I can't talk about its conformation. So with those sort of caveats about what you can't model aside, let's move into how you choose the rate function for your chemical reaction network. The first thing you should always use in a chemical reaction network is the mass action rate function. And this is because it's based in physics and has a very long history. I'm sure you've all seen these in high school, if not college chemistry courses. And so these rate functions were developed in the late 1800s by Peter Wage and Cato Maximiliano Goldberg. And they have this functional form. And this looks a little bit confusing, but I'll walk you through it. So each reaction has a rate, K sub R, and then we're going to multiply that reaction via this product operator by the number of inputs, by each input species raised to the power of how many that, of the stoichiometry of that species. So let's explain what this is. If I have nothing going to A, I have no input species, so my rate is just K. If I have A going to nothing at rate K, well, my input species here is A, so my rate is K times A raised to the power of 1 because only a single A is used in this reaction. If I have 2B plus C as my input species, my rate is going to be K times C, because there's one C that's being used, times B squared. So the stoichiometry of B is 2, so I raise B to the power of 2. And note that my output species don't matter. I only care about my input species here. And that gives me my mass action rate function. There are also non-mass action rate functions. And I really encourage you to think of these as approximations or model reductions of larger, more complex mass action chemical reaction networks. And a very commonly used example are Hill functions. So I'll show you these here because I think we will end up using them. A Hill function has this functional form and looks like the sigmoid curve, where it starts off low and gets high. And it has 3 parameters. Little k tells you what the maximum value is. So here we're raising little k, and it's raising the maximum value of my Hill function. The big K, which is down here and here, tells you where the inflection point is. So if big K is 5, my inflection point occurs around 5. If big K is 10, it occurs around 10. If big K is 20, it occurs over here. So that's sort of shifting the inflection to the right. And N determines how steep the Hill function is. So if N equals 1, you know, it's sort of quite gradual. But if N equals 4, I change very quickly from low to high values around my inflection point. And there's both positive Hill functions and negative Hill functions. Negative Hill functions go from their maximum value to 0, while positive Hill functions go from 0 to their maximum value. But they have the same sort of 3 parameters. Hill functions can be derived in some sort of limit from a more complicated mass action CRN. So I want to go through that derivation now so you guys get a flavor of how this works and understand why I think it's an approximation. I don't think I know it's an approximation. So let's start with this very traditional model of catalysis, where A binds to a catalyst and forms a complex, and that is then catalyzed to become B and released from the catalyst. This is called Michaelis-Menten kinetics, and I have 4 species and 4 reactions. I've written down the first 3 ODEs here, and I'm going to write down the 4th ODE here, and I'm going to make something called a steady state approximation. And so what I'm saying here is that the amount of A bound to the catalyst is not changing in time. So dAc dt is equal to 0. And this is called a steady state approximation because when something doesn't change in time, we say it's at steady state. This approximation is really related to the saturation of the catalyst by the substrate A. And you can derive it more rigorously in terms of that saturation if you want to. But I'm going to just say we're assuming things are at steady state. I can then add in this fact that I know my total amount of enzyme C is conserved. So the amount of C plus the amount of AC is equal to some constant C total. And when I take this equation and I combine it with the equation above and do a little bit of math algebra, I solve for my steady state concentration of the AC complex, which is equal to the total amount of C times this Hill function with the Hill coefficient of N. And here is what Km is. So my big K, I'm now calling it Km. I can then plug this function into my dynamics for B to get how much product my enzyme is producing. And notice that for every amount of product I produce, I have to degrade the same amount of A. So that's equal to the change, also the negative change in my substrate. That gives me this equation. So what I've done here is I went from initially having a mass action chemical reaction network with 4 species and 4 reactions, shown here, to a much simpler chemical reaction network that uses a Hill function, but only has 2 species and 2 reactions. So in some sense, I've gotten rid of the dynamics of the catalysts in this approximation. Before we break out into, or before we start looking at code, I want to sort of introduce a model that is worth seeing just to see how expressive mass action chemical reaction networks can be. And one of the problems in the workshop is to look at this from the lens of non-mass action kinetics. So this model, I'll just go through it. We have some signaling molecule that starts off external and can diffuse and become internal. It's called S. So S goes from external to S internal, which are these green guys. S can bind cooperatively to a transcription factor, which is inactive and becomes active when F binds to it. So we're using that binding motif. The transcription factor then can bind to a gene and form an active gene complex. An active gene can bind to a polymerase and form another complex. So we keep using this binding motif over and over again. When a gene binds to a polymerase, it can produce a transcript. This is similar to the catalysis I just showed you before. And there's a very simple model of transcription. A transcript can then bind to a ribosome and produce a complex, which, you guessed it, can be translated to produce a protein X. And finally, I'll let my transcript, my signaling molecule, and my protein be degraded or diluted, perhaps because we're really modeling a cell that is growing. And so I can write this out as a chemical reaction network. It has 10 reactions, more if you count the reversible ones as 2, and you can simulate it and get these trajectories of all the different species. And so this is really very similar to the type of systems many people study in the lab. And you can very succinctly describe your knowledge of the system that, S dimerizes as it binds to F in order to activate it in a chemical reaction network. So at this point, I would like to take some questions and go through a quick summary of what we've gone over. The first point is that chemical reaction networks are simulated as ordinary differential equations. The second point is that simple motifs like binding and catalysis can be reused over and over again to build complex networks of molecules interacting with each other. When you're confused about what rate function to use, you should always start using a mass action rate function because that comes from physics. And only use a non-mass action rate function to approximate more complicated mass action systems. And make sure you do that very carefully because a lot of people just sort of apply Hill functions left and right that aren't actually accurate in the regimes they're working in. So always go back and check to make sure that your approximation is correct. I'd like to acknowledge my co-instructors, Ayush, who you just heard, and Iran Agman, who will be teaching later. There's a bunch of us who've been building a lot of these tools. Biocompiler team is listed here, Zoltan, Andre, and Ayush, many of whom are going to be TAs today. We have many other TAs. They're all in the Zoom call. Maybe they can wave if they want to, but Ankita, Liana, Manisha, Matthew, Nasmus, Rory, Ryan, Sam, Andre and Zoltan are also TAing. You'll be seeing them on different days depending upon when people are available, but I would like to thank them all for taking their time to help with this. And finally, I'd like to thank Richard, Kate, and Drew, and all the other PIs who've made Build-a-cell a reality. And the NSF is supporting this series via their grant to Build-a-cell. And at the end of this, there are some selected references. If you want to learn more, feel free to take a look at the PowerPoint, which I put online.